Hello, everyone. My name is Adam. I am a MCAT expert and MCAT tutor at Shemmassian Academic Consulting. And today I'm going to walk you guys through a biology and biochemistry passage from the MCAT. All right, so to start out, let's break down kind of where we are in the MCAT and what our strategy is for the section as a whole. There's 4 sections of the MCAT. There's the chemistry and physics, then CARS, then biology and biochemistry, and psychology and sociology. Right, so this is the 3rd section, right? This is the 3rd out of 4 sections. This is the biology biochemistry passage. It comes right after the kind of 30 minutes lunch break. So strategy for the section as a whole. So we have 95 minutes and we have 10 passages and we have 15 discrete questions, right? So 10 passages, 15 discretes, right? So it makes the timing strategy fairly simple, right? We want to do the discrete questions in 1 minute each, right? That takes 15 minutes. And then 10 passages with now 80 minutes remaining. We want to do each of those in 8 minutes apiece, right? So that's kind of the goal for timing, to have like equal, I guess, effort towards each question. You want to kind of manipulate that a little bit as you begin practicing full-length exams as to how you should adjust these timers to what works best for your strengths in the exam. But yeah, 8 minutes and 8 minutes per passage, 1 minute per discrete question, and you're going to hit that 95-minute mark with giving yourself kind of adequate time for each question. So that's kind of the basic strategy. So now that we're in the section, let's say that we've done our discretes, we've come across this passage. And so we're going to break down kind of the most basic strategy for a passage, which is you read the passage itself, right? So we're going to read these paragraphs, kind of analyze the figures briefly, and then answer the questions, right? So remember, we have 8 minutes total to do this. We note as we just look at this that there is 5 questions, right? So we're going to be doing 5 total questions. So basically we want to do kind of about 3 minutes to analyze the passage and kind of get a gist, and then about a minute per question, and we should be able to hit that in about 8 minutes. I'm not going to be doing that today because I'm going to be talking through my kind of thought process as I'm doing it, but that when you're practicing on your own, that's the goal we're shooting for. All right, so let's get it started here. So granulocyte colony stimulating factor receptor, GCSFR, right? So I'm going to highlight GCSFR, right? Because that's an acronym that we may or may not see show up in the questions. We want to be able to find it in case we have to refer back. So it's a transmembrane cytokine receptor involved in the production of neutrophil granulocytes. Okay, this is all more definition on what GCSFR is. Mutations in these receptors have been discovered in cancers such as acute myeloid leukemia, AML, and chronic neutrophilic leukemias, right? These are 2 different forms of leukemia, right? Both acronyms. Let's highlight them in case they show up. I'm also going to highlight the word mutation and the word cancer, right? Because those should kind of ring off as bells in your brain when you see those 2 words, because those are things that we should know for the MCAT. We should be familiar with how kind of cancer works roughly, and mutations in genes and stuff like that create stuff. Transcription, translation, protein, stuff like that. Mutations in the DNA. That's a huge MCAT related concept. The aberrant signaling mechanisms of GCSFRs, however, are poorly understood, right? So what does the word aberrant mean? It means like not working as functioning, or like the abnormal, right? So the aberrant signaling, the ones that's not signaling properly, either more or less, depending on what that's working. So we don't know how that works. So we go into the next paragraph here. Scientists hypothesize, so they're trying to figure out how it works, that understanding the downstream protein targets of the mutant GCSFRs might provide information about how these GCSFRs contribute to cancer. Okay, so we're going to be looking at downstream protein targets, right? So what that kind of means is we have sort of like, let's draw it out. we have a sort of like stream, right? So we have a receptor, right? It's going to be on our membrane. Our receptor, we're going to have our intracellular, extracellular, and transmembrane portions, right? They mentioned it's a transmembrane cytokine receptor. And we're looking at this receptor and we have downstream protein targets, which means like we're going to have something happening, a cascade. Things are going to other things, a cascade. We don't actually know much about how this cascade works, but then we have some form of protein down here that's a target for something. We'll figure out what that is later, but that's kind of what that means. That's what they're looking at. The researchers developed an assay to detect levels of p-STAT5 and p-ERK1/2 in the presence or absence of GCSF. Okay, so now we're getting into all these like wordy acronyms and stuff. So let's keep it simple. What is this stuff? p-STAT5, we can assume that's probably a protein, and P probably means phosphorylated. That's a pretty common format that you'll see a lot of the times in these passages, is that P put before the name of a protein to mean it's phosphorylated, and P-ERK1, so two phosphorylated proteins. And then presence or absence of GCSF, right? Notice we don't have the R there, which means this would be like the ligand for the receptor. And we're like, okay, what kind of ligand is this? They told us at the top that it was a cytokine, right? Cytokine receptor. So the cytokine must be the ligand for the receptor. So GCSF would be that cytokine. For the wild type GCSFR, that's what WT stands for, wild type. And then T618I mutant GCSF. Okay, so this format, T618I, is a very common format that we see a lot on the MCAT. So I'm going to write it down here. But if we have G, or no, we don't have G. Let's erase that. And we have T618I. It is a good idea to get familiar with this type of format because it comes up all the time on MCAT passages. T, that is the amino acid that was initially, right? So this would be the wild type amino acid. 618 is the position, right? That's the position at which we have this particular amino acid. And then I is the substitution, right? Or the mutation, right? So we have mutated a T, which is an amino acid, right? That's threonine. And then for I, which is isoleucine, right? So that's going to be a big change, right? Because, right, threonine is polar and can be phosphorylated, and isoleucine is non-polar and can't be phosphorylated. So we're changing groups altogether and probably changing the functionality. So this is a common format. It's a good idea to be familiar with this type of format. Okay, so that's what we're looking at. We're looking at this one mutant versus the wild type. Let's move on in the passage here. So now we have a figure. The results are shown in figure 1. So what we want to do with this figure is we want to break it down briefly with tide P. So what does tide P mean? I'm going to write it over here. TAI, D, and P, right? So T meaning title, A meaning axes, I meaning independent variable, right? So I'm going to say IV for independent variable, D for dependent variable, and then P for pattern. What type of patterns do we see in our figure, whether it's a graph or a table or whatever? So we want to try and pick up on these things, right? Again, we're limited by time, so we don't want to go too deep into it, but we do want to have a gist for all of these different things to kind of get an understanding. All right, so let's look. Title, right at the bottom here, we'll see figure 1, and they gave us the title, target phosphorylation of proteins, right? We kind of expected that with the p-STAT and the p-ERK. Target phosphorylation of proteins under different conditions. Okay, now let's look at our axes. Across the top here, we got GCSF, wild type, and our mutant, as we would expect. What's our independent variable? It looks like we are changing the concentration of our cytokine, GCSF, right, which is our ligand, right? So we go from none, right, that's the presence or absence thing, and then a certain amount of time, right, 12.5 minutes and 90 minutes, and then we have the same thing down here. and they're looking at the two different things, right? And then dependent variable is all of these little spots in here, which is showing us levels of phosphorylation for the wild type or the mutant for these different proteins, and whether or not they're present or not. And then patterns. So what's happening here? The wild type, nothing, 12.5 minutes. We have a lot, then less. It's kind of the same thing twice in a row for both of these different proteins. Then for the mutant, we have a little bit less, right? You notice the difference here. There's a difference between wild type and that, and then a little bit greater. So it doesn't phosphorylate as much, but then it stays phosphorylated longer. That's interesting. And then do we see the same trend on ERK? No, we don't. You notice the ERK looks almost identical to the wild type for the mutation. So it doesn't seem to impact that at all. So that's kind of interesting to note. So we went through Tide P, just kind of paid attention. You could write down on your scratch paper if you'd like for this. You can also just kind of go through it and use that. Maybe a little bit of highlighting. It's hard to always highlight on figures. Sometimes you can't. So sometimes it's good to write it down on your scratch paper. All right, now let's finish out the last paragraph. In subsequent experiments, the researchers tested other mutants, okay, including Q741N, right? There's that same format of at position 741, used to be Q, now it's N, which it's lowercase n, so that means something else, where N indicates any of the other 19, so anything except for Q, any of the other 19 amino acids. They found that some substitutions led to increase, while others decreased all signaling activity. In the future, the researchers hope to find new downstream targets of cancer-relevant genes. So this is not a super helpful paragraph. Just they tried other things, and some helped, some didn't, some increased, some decreased. So maybe let's highlight that just in case they do ask us a question, but it doesn't seem like a great paragraph that's going to be super useful for questions. Okay, so ideally you want to just have done that whole process in about 3 minutes. Obviously that took way longer than that, but that's the goal for that. Now let's do the questions. Again, we're aiming for about a minute per question using the information that we just got there. All right, number one. One researcher hypothesizes that these GCSFRs activate the MAP kinase pathway. Which of the following protein residues is most likely to be altered by this pathway? All right, so this is a very interesting question, right? Because we almost don't need the passage for this, right? Because we have this thing and it activates a pathway, right? This would be kind of that downstream idea. And what type of pathway? It's a kinase pathway. So we're using our outside knowledge for what is a kinase, right? Kinases phosphorylate things, right, from ATP, right? So that's similar to a phosphorylase where we phosphorylate things from inorganic phosphate, but kinases phosphorylate things from ATP. So we need to be able to be phosphorylated. What type of residue, or another word for amino acid, which of the following residues, which amino acid would be altered? Which of these amino acids can be phosphorylated? So tryptophan, right, that's amino acid W, which is not going to be able to be phosphorylated, right? Because what type of amino acid is that? That is aromatic and nonpolar with no phosphorylatable residues. So not going to be A. Arginine, right, that's going to be charged, right? We call that amino acid R. That's going to be charged. Which way is it charged? It's charged positive, which means it's basic. So it's positive, basic. And that also cannot be phosphorylated. And then glutamate, that's also going to be charged, right? That's going to be negatively charged, right? And so glutamate is E, right? For glutamate or glutamic acid. Negatively charged or acidic. And then serine, that's S, and that's going to be polar, uncharged. Which of these can be phosphorylated? Only serine, right? What are our other amino acids that can also be phosphorylated? There's only 3 of them in eukaryotes. It's going to be either serine, so I'm going to actually write it over here. The 3 that can be phosphorylated are serine, threonine, and finally tyrosine, right? So they all have that OH or hydroxyl group there on the end, our alcohol group, that allows our phosphate, our PO4, to attach, right? So we're looking for any of these threes. These are the three in eukaryotes, right? And then again, if we go over to prokaryotes, we add one additional amino acid, histidine, that can be phosphorylated in prokaryotes. But in eukaryotes, only S, T, and Y. So there it is, S. Cool, pretty straightforward. Number two, what is the effect of the mutation, the T618 mutation on pStat5 activation based on the results from figure 1? Okay, so we're going to look at figure 1. Right, so let's look at the mutation on pStat5 on figure 1. What do we have going on here? Right, we're looking at phosphorylation, right? We have our two trend points here. We kind of highlighted them there. They're the same when nothing's going on. After 12 minutes, wild type is more phosphorylated. After 9 minutes, it's less. T618 doesn't do as much and then kind of stays the same, but is greater than wild type. So we're looking for some kind of trend along those lines. So A, the mutant shows less phosphorylation at both time points when compared to wild type. We know that that is not true because it's less and then more. So that's just a false statement. Not going to be the correct answer. The T618I shows less phosphorylation at only the early time point when compared to wild type. That's true, right? The early time point we got less, and then we have more. So only the early. So that one's true. So we're going to hold that one as probably the correct answer. Let's move to C. The 618I mutant decreases the binding affinity of the ligand to the protein. I'm not sure why the ligand's attaching to the protein, right? Because remember, the ligand's attaching to the receptor, which then later would phosphorylate the protein. So that's not even the correct kind of connection there. Also, the graph doesn't show us, or the figure doesn't show us binding affinity. The figure is showing us phosphorylation, right? So this term of binding affinity is, we can't evaluate that. We don't know. So we're going to take that one out. And then last year, the T618I mutant shows no decrease in binding. Again, we're talking about binding affinity. We're not talking about that in the figure. That wouldn't be answering our question at all, right? This could be a true statement though, right? The T618 mutant shows no decrease in binding affinity, right? Showing no decrease. I mean, we don't know anything about it. So it doesn't really show that. So it's actually not a true statement. The figure does not show us that we have no decrease, and we don't know anything about it, right? So A, C, and D are all bad answers. So we would have to go with B here. B is a true statement. Less at only the early and not the later. That was true. So let's B. All right, and the answer choice, or number 3 here, which amino acid residue is more likely to be found in the transmembrane region of the mutant? Well, I guess it doesn't really matter, because in the transmembrane we're not having a lot of activity in the transmembrane because it's just in the middle, right? And if we go back over to our little picture of what our receptor looks like, right? We got extracellular, we got intracellular, and then we got the transmembrane, right? And what does our membrane look like, right? It's got, what do we call this again? A phospholipid bilayer, right? So on the tops, or kind of on these parts here, these are going to be our phosphate groups, right? Because they're polar. They're going to be close to our water out here, right? Because they're hydrophilic, and they're polar because water is very polar. Hydrophilic residues are polar. So our phosphates on here are polar. And then we have our little chains in our bilayer coming out, right? Again, we also would have phosphate here, polar down here as well. And then our chains, which come together, and in the middle here, we're going to be hydrophobic, right? We're as far as we can from water, we're kind of by each other, right? So we're going to be very hydrophobic in the middle here, or non-polar. We can call ourselves non-polar or hydrophobic. So we're looking for something that's in the transmembrane region, which is this middle. We're looking for something non-polar. We're going to look for a non-polar amino acid. All right, right away, A, alanine. We know alanine is non-polar. We could just answer with A because we know that that is true. Let's go through for the sake of practice and look at the other ones and make sure that they're all incorrect. Threonine, right? That's one that we talked about earlier. That can be phosphorylated. That's polar, right? So polar, not good. X. Aspartate, right? Charged, negatively charged. right? Not good. And then lysine, polar, right? Oh, sorry, that's not polar. That's charged. It's one of our charged residue. Positive, right? It's going to be, yeah, it's going to be one of our charged, positively charged residues. So interesting, we have a non-polar, a polar, a negatively charged, and a positively charged residue. We're kind of covering all of the different categories there. Right, we're looking for the transmembrane one, which is alanine, the non-polar uncharged. And then figure 1, so yeah, 3 is A. 4, figure 1 demonstrates that in the absence of UCSF, which again, that's our ligand or our cytokine, the receptor cannot do what? So in the absence, right, let's go look at the figure. In the absence, we have no phosphorylation downstream. So we cannot phosphorylate downstream. So we got to look for an answer with that. So A, directly phosphorylated target. Well, it can never do that, regardless of absence or presence, because that's not something that it does. So it's probably not a great answer. Kind of technically a true statement. It does not directly phosphorylated targets, but it doesn't really answer our question because we're talking about the absence of GCSF. This is not related to that, even though it's kind of a true statement. It does not directly phosphorylated targets. And then B, activate downstream signaling to phosphorylated target. That looks a little bit better, right? Because that's what we are not doing. So that is related to the absence. So that's a better answer. Stick with that one for now. C, produce neutrophil granulocytes, right? We talked about production of neutrophil granulocytes at the beginning here, but again, that's not part of our figure 1. We're talking about downstream phosphorylation in figure 1. We don't really know exactly how that relates to production neutrophil granulocytes. We kind of assume that it's correlated, but they didn't outline that for us in the passage. So we're going to take that one out as being kind of outside of the scope. We don't know that from figure 1. We'd have to get some additional information. And then D, suppress p-STAT5 or p-ERK transcription. That's a little bit of a weird word to use there because, right, transcription is the process of creating RNA from DNA, right? The transcription is the process making this. And right, p-STAT5 and p-ERK1/2 are proteins, right? So we would have to translate RNA to make the protein, and then they're post-translationally modified. That's what the P is, right? That's phosphorylated. So they're post-translationally modified proteins. So the word transcription here is not a great term. So also we're talking about phosphorylation, which is post-translational modification. So not a great answer there. Also, right, we're talking about this, right? So we're not suppressing transcription. Not great answer. So B is the only one that really makes any sense here. So we're gonna go with B. And then our last question here. A researcher is attempting to understand the temporal trend, right? So temporal means time, observed in p-STAT5 or p-ERK1/2 phosphorylation. We use RT-qPCR, right? Reverse transcription, quantitative, real-time PCR, which we're looking at mRNA when we're talking about that, right? mRNA is kind of one of the big things for RT-PCR. Might show upregulation, right? That's like an increase in activity in which type of enzyme. Okay, so in p-STAT5 and PRK1/2, and we're looking at temporal trend. So over time, so first we introduce it, we have a lot of phosphorylation, and over time we decrease phosphorylation. That's true for both of them. Right away we add it, a lot of phosphorylation. We're continuing to add it. Over time, we have a decrease in phosphorylation. Right, we talked about earlier kinases and phosphorylases use either ATP or inorganic phosphate. Right, kinase uses ATP, phosphorylases use inorganic phosphate. But we're doing the opposite here, right? We're taking away phosphate groups, so we're dephosphorylating, which is what a phosphatase does. Right, so we're looking for that. It is answer choice B, so that is fairly easy to just answer if we wanted to just straight up answer, but let's talk about the other ones as well as to why they're not true, because what are these other types of enzymes? A transferase is going to be transferring one group from another. Do we see an increase in transferring of one group for another? Like, no, we don't. We don't really see any of that. We're seeing a decrease in phosphorylation. So transferate, we're not transferring one group for another. We are seeing a decrease in phosphorylation. So that's a good answer. Oxidoreductase, right? That's a type of enzyme that's going to change our oxidation state of a particular atom, right? An example of a redox reaction might be like H2 plus O2, right? A form of peroxidase, right? H2O2, right? This is a redox reaction, right? Because this is neutral. This is neutral in their diatomic state. And then all of a sudden, hydrogen becomes +1, oxygen becomes -2, right? Which one's being oxidized, which one's being reduced, right? You can use our mnemonic. Leo goes Gur, right? So losing electrons, oxidation, gaining electrons, reduction, right? So who's losing electrons? Hydrogen, right? Hydrogen went from 0 to +1. We lost an electron. So hydrogen is oxidized, and then oxygen gained 2 electrons, went from neutral to -2, therefore reduced. So oxygen was reduced, right? So that we would use an oxidoreductase for that. We don't have that. And then ligase would be, ligases ligate things, right? They combine them. So a ligase will combine 2 different things. We don't really have combination of 2 things at all in the entire passage, right? That would be like A plus B. And then we have a ligase that's going to make AB. Yeah, that would be a good example of it. We don't have that. So yeah, the answer is going to be B. I hope each of those questions made sense. Yeah, go ahead and put some questions in the comments. And yeah, thank you for following along with me in this passage. We have a daily passage that can be sent to your inbox every day. It's going to be in the transcript, or in the transcription, in the description below. And you can click that link and add your email to the list, and we'll send you a daily passage every day. So yeah, thank you.